Molecular basis of inheritance. 1. RNA mostly function as a messenger, structural adapter. Messenger. 2. DNA is the most abundant genetic material. False. 3. DNA is a polymer of deoxyribonucleotides. 4. No. Of nucleotides in O174 is 5386. 5386. 5. O174 is which type of virus? Bacteriophage. 6. Total bases in bacteriophage lambda is 97,014 bases. 97,014 bases. 7. Dash has 4.6. x 10⁻6 BP. E. coli. 8. Diploid content of human DNA is dash bases. 13.2. x 10⁻9. 13.2. x 10⁻9. 13.2. x 10⁻9. 9. Three components of nucleotides are pentose sugar, nitrogenous bases, phosphate group. Pentose sugar, nitrogenous bases, phosphate group. Pentose sugar, nitrogenous bases, phosphate group. 10. Thymine is present in RNA. False. 11. Nitrogenous base is linked to the O of L, C, 5'C of pentose sugar through dash linkage. N-glycosidic linkage. 12. Deoxythymine is present in DNA. True. 13. Thymine and uracil have differences in a dash group. Methyl. 14. In RNA, every nucleotide residue has an additional O group present at dash position in the ribose. 2 feet. 15. In RNA, dash is present in place of thymine. Uracil. 16. Thymine other chemical name is 5-methyl uracil. 17. DNA was first identified by DASH as an acidic substance in year dash. Friedrich Miescher, 1869. 18. He named DNA as nuclein. 19. X-ray differentiation data of DNA was produced by the scientists Morris Wilkins and Rosalind Franklin. 20. Double helix model of DNA was proposed in year dash by dash n. 1953, Watson and Crick. 21. One of the hallmarks of their proposition was base pairing between the two strands of DNA. True. 22. Dash observed that ratios between adenine and thymine and guanine and cytosine are constant. Irwin Chargaff. 23. Bases are paired through covalent bond. False. 24. Adenine form _no of H bond and guanine form -no of H bond. 2, 3. 25. Generally DNA helix is coiled in a right-hand fashion. True. 26. The pitch is -nanometer and -no of base pairs are present in each turn. 3.4. Nanometer, 10 to 3.4. 3.4. Nanometer, 10. 3.4. Nanometer, 10. 27. The plane of one base pair stacks over the other in double helix. This, in addition to H bonds, confers stability to the helical structure. True. 28. Dash proposed the central dogma of molecular biology. Francis Crick. 29. No of bases between first and 15th base pair of a DNA helix are distance between them is 26, 14 by 3.4. x 10⁻10. 26, 14 by 3.4. x 10⁻10. 26, 14 by 3.4. x 10⁻10. 30. Length of a DNA molecule is 1 micrometer. What will be the no of base pairs in it? 0.033 times 10 to the 9 BP. 0.033 times 10 to the 9 BP. 0.033 times 10 to the 9 BP. 31. The length of human DNA is 2.2 m diploid. 2.2 m diploid. 2.2 m diploid. 32. Length of E. coli DNA is 1.36 millimeters. 1.36 millimeters. 1.36 millimeters. 33. DNA in nucleoid is organized in large loops held by proteins. True, true, true. 34. Histones are rich in amino acids dash and dash. Arginine and lysine. Arginine and lysine. Arginine and lysine. 35. Both amino acid residues carry positive charges in their main chains. False, inside chain. False, inside chain. False, inside chain. 36. Histone octamer contain proteins. 2 copies of H2A, H2B, H3 and H4 out of NCERT. 2 copies of H2A, H2B, H3 and H4 out of NCERT. 2 copies of H2A, H2B, H3 and H4 out of NCERT. 37. Identify the structure shown in diagram. I. Label A, B, C and D. Nucleosome, A, DNA, B, H1 histone. C. Histone octamer, D. Core of histone molecules. Nucleosome, A, DNA, B, H1 histone. C. Histone octamer, D. Core of histone molecules. Nucleosome, A, DNA, B, H1 histone. C. Histone octamer, D. Core of histone molecules. 38. What is a nucleosome? Negatively charged DNA is wrapped around the positively charged histone octamer. Negatively charged DNA is wrapped around the positively charged histone octamer. Negatively charged DNA is wrapped around the positively charged histone octamer. 39. A typical nucleosome contains -BP of DNA helix. 200, 200, 200, 40. Function of non-histone chromosomal NHC proteins is packaging of chromatin at higher level, packaging of chromatin at higher level. Packaging of chromatin at higher level. 41. Chromatin that is more densely packed and stains dark are called heterochromatin, heterochromatin, heterochromatin. 42. Loosely packed and stains light is called dash euchromatin, euchromatin, euchromatin. 43. Heterochromatin is transcriptionally active. False, false, false. 44. In transforming principle, dash did a series of experiments on dash. Frederick Griffith, Streptococcus pneumoniae. Frederick Griffith, Streptococcus pneumoniae. Frederick Griffith, Streptococcus pneumoniae. 45. S-strain bacteria have dash coat. Mucus polysaccharide, mucus polysaccharide, mucus polysaccharide. 46. Living S bacteria can be recovered from a solution where S bacteria may not be present. True, true, true. 47. Genetic material was earlier thought to be RNA, protein. Protein, protein, protein. 48. Dash, dash, dash concluded that DNA is the hereditary material. Oswald Avery, Colin MacLeod and Maclean McCarty. Oswald Avery, Colin MacLeod and Maclean McCarty. Oswald Avery, Colin MacLeod and Maclean McCarty. 49. Dash and dash gave the unequivocal proof that DNA is genetic material in year dash, Alfred Hershey and Martha Chase, 1952. Alfred Hershey and Martha Chase, 1952. Alfred Hershey and Martha Chase, 1952. 50. Hershey and Chase used which bacteria? E. coli, E. coli, E. coli. 51. Virus particles were separated from the bacteria by spinning them in centrifuge. Spinning them in centrifuge. Spinning them in centrifuge. 52. Viral coats were removed from the bacteria by agitating them in blender. Agitating them in blender. Agitating them in blender. 53. Dash isotope of S and dash isotope of P were used in Hershey-Chase experiment. 35S, 32P. 35S, 32P. 35S, 32P. 54. The 3 steps of Hershey-Chase experiment were infection, blending and centrifugation. Infection, blending and centrifugation. Infection, blending and centrifugation. 55. QB bacteriophage have DNA as a genetic material. False, false, false. 56. TMV have RNA as a genetic material. True, true, true. 57. Criteria for a molecule which act as genetic material are criteria to act as genetic material. I. Replication. E. Stable chemically and structurally. E. Provide scope for mutation. IV. It should be able to express itself in the form of Mendelian characters. Criteria to act as genetic material. I. Replication. E. Stable chemically and structurally. E. Provide scope for mutation. IV. It should be able to express itself in the form of Mendelian characters. Criteria to act as genetic material. I. Replication. E. Stable chemically and structurally. E. Provide scope for mutation. IV. It should be able to express itself in the form of Mendelian characters. Criteria to act as genetic material. I. Replication. E. Stable chemically and structurally. E. Provides scope for mutation. IV. It should be able to express itself in the form of Mendelian characters 58. Stability is a property of genetic material was evident from dash experiment. Griffiths. 59. Tell the two differences between DNA and RNA. I. DNA don't have 2 O group on sugar. E. DNA have thymine in place of uracil. I. DNA don't have 2 O group on sugar. E. DNA have thymine in place of uracil. I. DNA don't have 2 O group on sugar. E. DNA have thymine in place of uracil 60. The protein synthesizing machinery has evolved around RNA, DNA. RNA. 61. 2 O group present at every nucleotide in RNA is a reactive group and makes RNA labile and easily degradable. True. 62. Presence of thymine at the place of uracil do not confer any stability to DNA. False. 63. RNA mutate at a slower rate. False, faster. 64. Protein was the first genetic material. False, RNA. 65. DNA resists change by evolving a process of repair. True. 66. DNA replicated conservatively. False. 67. Semi-conservative model was proved by dash and dash in year dash. Matthew Meselson and Franklin Stahl, 1958. Matthew Meselson and Franklin Stahl, 1958. Matthew Meselson and Franklin Stahl, 1958. 68. N15 radioactive isotope was used in their experiment. False, N15 is not a radioactive isotope, it is a heavy isotope of N. False, N15 is not a radioactive isotope, it is a heavy isotope of N. False, N15 is not a radioactive isotope, it is a heavy isotope of N. 69. Heavy DNA molecule could be distinguished from normal DNA by centrifugation on dash gradients. CSCl. 70. E. coli divided in DASH-min. 20. 71. Taylor performed experiments on dash using dash in year dash and showed that Vischer Faba beans, radioactive thymidine, 1958, DNA replicates semi-conservatively. Vischer Faba beans, radioactive thymidine, 1958, DNA replicates semi-conservatively. Vischer Faba beans, radioactive thymidine, 1958, DNA replicates semi-conservatively. 72. Taylor performed experiments to know whether DNA replicated semi-conservatively or not. False. 73. In E. coli, average rate of polymerization has to be approximately -BP per second. 2000. 74. Energetically replication is an expensive process. True. 75. In addition to acting as substrates, also provide energy for polymerization reaction. Deoxyribonucleotides triphosphates. 76. In long DNA, replication occurs within a small opening of DNA helix, referred to as replication fork. 77. Discontinuous synthesis occurs in template strand with polarity 5-3. 78. Discontinuously synthesized fragments are later joined by which enzyme? DNA ligase. 79. DNA polymerases initiate the process of replication. False, it is initiated by RNA primer. False, it is initiated by RNA primer. 80. Replication initiates randomly at any place in DNA. False. 81. There is a definite region in E. coli DNA where the replication originates. Such regions are called dash. Origin of replication. 82. A piece of DNA, if needed to be propagated during recombinant DNA procedures, requires a dash because it provides the origin of replication. Vector. 83. Enzymes used in DNA replication are helicase, DNA polymerase, primase, DNA ligase, topoisomerase out of NCERT. Helicase, DNA polymerase, primase, DNA ligase, topoisomerase out of NCERT. Helicase, DNA polymerase, primase, DNA ligase, topoisomerase out of NCERT. 84. Dash fragments are formed in the lagging strand. Okazaki out of NCERT. Okazaki, out of NCERT. Okazaki, out of NCERT. 85. A schematic structure of a transcription unit is shown in diagram. 2. Identify the strands X and Y in the regions A, B and C. X, template strand, Y, coding strand, A promoter, B structural gene, C terminator. X, template strand, Y, coding strand, A promoter, B structural gene, C terminator. X, template strand, Y, coding strand, A promoter, B structural gene, C terminator. 86. The enzyme that help in opening of DNA helix during transcription is RNA polymerase. RNA polymerase. RNA polymerase. 87. The transcription unit consists which three of regions? Promoter, structural gene and terminator. Promoter, structural gene and terminator. Promoter, structural gene and terminator. 88. Transcription happens at the coding strand. False, it happens at the template strand. False, it happens at the template strand. False, it happens at the template strand. 89. Template strand polarity is 3, minus 5, 3, minus 5, 3, minus 5, 3, minus 5, 90. All the reference point while defining a transcription unit is made with template strand. False, it's made with coding strand. False, it's made with coding strand. False, it's made with coding strand. 91. Coding strand polarity is 5, minus 3, 5, minus 3, 5, minus 3, 92, 5, end of template strand means upstream. False, 5, end of coding strand means upstream. False, 5, end of coding strand means upstream. False, 5, end of coding strand means upstream. 93, 3 feet end of coding strand means downstream. True, true, true. 94. Presence of a promoter in a transcription unit defines template and coding strands. True, true, true. 95. Dash is the functional unit of inheritance. Gene, gene, gene. 96. What is cistron? A segment of DNA coding for a polypeptide, whereas gene codes for everything. A segment of DNA coding for a polypeptide, whereas gene codes for everything. A segment of DNA coding for a polypeptide, whereas gene codes for everything. 97. Structural gene is monocistronic in prokaryotes, eukaryotes. Eukaryotes, eukaryotes, eukaryotes. 98. The actual coding or expressed sequences are called dash. Exons, 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 exons. 99. Inheritance of a character is never affected by promoter and regulatory sequences of a structural gene. False, false, false. 100. Regulatory sequences are sometimes loosely defined as regulatory genes, as these sequences code for RNA. False, false, false. 101. Name the three types of RNA in prokaryotes: mRNA, rRNA, tRNA, mRNA, rRNA, tRNA, mRNA, rRNA, tRNA. 102. mRNA is found in the highest amount in comparison of other rRNA. False, rRNA is found in highest amount 80%. False, rRNA is found in highest amount 80%. False, rRNA is found in highest amount 80%. 103. The smallest RNA among all the three is tRNA, tRNA, tRNA. 104. Dash plays structural and catalytic role during translation. rRNA, rRNA, rRNA. 105. How many RNA polymerases found in bacteria and name them? 1. DNA-dependent RNA polymerase. 1. DNA-dependent RNA polymerase. 1. DNA-dependent RNA polymerase. 106. RNA polymerase binds to dash and initiates transcription. Promoter, promoter, promoter. 107. It uses nucleotide triphosphates as substrate. False, it uses nucleoside triphosphate not nucleotide triphosphate. False, it uses nucleoside triphosphate not nucleotide triphosphate. False, it uses nucleoside triphosphate not nucleotide triphosphate. 108. Helicase facilitates opening of helix and continues elongation during transcription. False, RNA polymerase do this thing. False, RNA polymerase do this thing. False, RNA polymerase do this thing. 109. Initiation factor is dash and termination factor is dash. Sigma, rho, sigma, rho, sigma, rho. 110. Transcription and translation can be coupled in dash. Bacteria, bacteria, bacteria. 111. In eukaryotes, how many RNA polymerases are present in nucleus? 3, 3, 3. 112. 5 srRNA is synthesized by RNA polymerase 3. RNA polymerase 3. RNA polymerase 3. 113. Tell the functions of all the 3 RNA polymerases. RNA polymerases transcribes, I. RNA polymerase 1 rRNA, 28s, 18s and 5, 8s, E. RNA polymerase 2, hnRNA, E. RNA pol. 3. Terna, 5 srRNA, and snRNA. RNA polymerases transcribes, I. RNA polymerase 1 rRNA, 28s, 18s and 5, 8s, E. RNA polymerase 2, hnRNA, E. RNA pol. 3. Terna, 5 srRNA, and snRNA. RNA polymerases transcribes, I. RNA polymerase 1 rRNA, 28s, 18s and 5, 8s, E. RNA polymerase 2, hnRNA, E. RNA pol. 3. Terna, 5 srRNA, and snRNA. 114. What is hnRNA? Nascent RNA containing both introns and exon. Nascent RNA containing both introns and exon. Nascent RNA containing both introns and exon. 115. In capping dash is added at 5' end of hnRNA, methylguanosine triphosphate, methylguanosine triphosphate, methylguanosine triphosphate. 116. In tailing dash to dash no of dash residues are added in a template dependent, independent manner. 200 to 300, adenylate, independent. 200 to 300, adenylate, independent. 200 to 300, adenylate, independent. 117. The presence of exons is reminiscent of antiquity. False. Introns. False. Introns. False. Introns. 118. The process of splicing represents the dominance of dash. RNA world. RNA world. RNA world. 119. Who was a physicist, biologist, first proposed that codons should be triplet? George Gamow, physicist. George Gamow, physicist. George Gamow, physicist. 120. The chemical method developed by Dash was instrumental in synthesizing RNA molecules with defined combination of bases. Har Gobind Khorana. Har Gobind Khorana. Har Gobind Khorana. 121. Whose cell-free system for protein synthesis finally helped the code to be deciphered? Marshall Nirenberg's. Marshall Nirenberg's. Marshall Nirenberg's. 122. Dash enzyme was helpful in polymerizing RNA in a template-independent manner. Severo Ochoa. Severo Ochoa. Severo Ochoa. 123. Which amino acids have only I codon? Tell the codon. Methionine, August, tryptophan, UG. Methionine, August, tryptophan, UG. Methionine, August, tryptophan, UG. 124. Which 9 amino acids have 2 codons? ASN, ASP. CYs, GLN, GLU, HIS, LYS, Phi, TYR. ASN, ASP. CYs, GLN, GLU, HIS, LYS, Phi, TYR. ASN, ASP. CYs, GLN, GLU, HIS, LYS, Phi, TYR. 125. Which amino acid has 3 codons? Isoleucine. 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 126. The 4 codons of threonine, proline, valine, alanine and glycine are: threonine, AC is constant equals ACU, ACC, ACA. ACC, proline, CC is constant equals CCU, CCC, CCA, CCG. Valine GU is constant equals GU, GUC, GUA, GUG. Alanine, GC is constant equals GCU, GCC, GCA, GCG. Glycine, GG is constant equals GGU, GGA, GGC, GGG. Threonine, AC is constant equals ACU, ACC, ACA. ACC, proline, CC is constant equals CCU, CCC, CCA, CCG. Valine GU is constant equals GU, GUC, GUA, GUG. Alanine, GC is constant equals GCU, GCC, GCA, GCG. Glycine, GG is constant equals GGU, GGA, GGC, GGG. Threonine, AC is constant equals ACU, ACC, ACA. ACC, proline, CC is constant equals CCU, CCC, CCA, CCG. Valine GU is constant equals GU, GUC, GUA, GUG. Alanine, GC is constant equals GCU, GCC, GCA, GCG. Glycine, GG is constant equals GGU, GGA, GGC, GGG. 127. Name the codons of acidic amino acids. Ga is constant equals GO, GAC for aspartic acid and GA, GAG for glutamic acid. Ga is constant equals Go, GAC for aspartic acid and GAG for glutamic acid. Ga is constant equals Go, GAC for aspartic acid and GAG for glutamic acid. 128. Stop codons are UAA, UGA, UAG. UAA, UGA, UAG. UAA, UGA, UAG. 129. Which three amino acids have 6 codons? Serine, arginine, leucine, mnemonic, LAS. Serine, arginine, leucine, mnemonic, LAS. Serine, arginine, leucine, mnemonic, LAS. 130. The two codons that codes for lysine are AAA AG, AAA AG, AAA AG. 131. Dash act as a initiator codon and is the only codon for the amino acid EUG, methionine. EUG, methionine. EUG, methionine. 132. What does degeneracy of codon mean? Many codons code for single amino acid. Many codons code for single amino acid. Many codons code for single amino acid. 133. What does unambiguity of codon means? One codon code for only one amino acid, one codon code for only one amino acid, one codon code for only one amino acid. 134. The codon is read in mRNA in a contiguous fashion. There are no punctuations. True, true, true. 135. Genetic code is nearly universal. True, true, true. 136. Exceptions of the universality of genetic code is found in mitochondrial codons and some protozoans. Mitochondrial codons and some protozoans. Mitochondrial codons and some protozoans. 137. Tell all the features of genetic code. Degeneracy unambiguous, universal read in contagious fashion. Degeneracy unambiguous, universal read in contagious fashion. Degeneracy unambiguous, universal read in contagious fashion. 138. Mutation can result in a gain of function. True, true, true. 139. Insertion of one or two bases changes the reading frame from the point of insertion. Such mutations are called insertion mutations. False, called frame shift mutation. False, called frame shift mutation. False, called frame shift mutation. 140. Identify the structure shown in diagram. 3. Identify A, B, C and X, Y, Z. Diagram 3A cell, BTYR, CMRNA. X terna, Y anticodon, Z codon. Diagram 3A cell, BTYR, CMRNA. X terna, Y anticodon, Z codon. Diagram 3A cell, BTYR, CMRNA. X terna, Y anticodon, Z codon. 141. The tRNA was earlier called dash. Sarna, soluble RNA, Sarna, soluble RNA, Sarna, soluble RNA. 142. Dash loop of terna has bases complementary to anticodon, anticodon, anticodon. 143. There is a specific terna for initiation called as dash. Initiator terna, initiator terna, initiator terna. 144. Describe the four ends of terna and their functions. Four ends of terna: I, glue loop, ribozyme binds here. E, anticodon loop, mRNA binds here. E, TC loop, ribosome binds here. IV, CCA sequence, amino acid attaches here. Four ends of terna: I, glue loop, ribozyme binds here. E, anticodon loop, mRNA binds here. E, TC loop, ribosome binds here. IV, CCA sequence, amino acid attaches here. Four ends of terna: I, glue loop, ribozyme binds here. E, anticodon loop, mRNA binds here. E, TC loop, ribosome binds here. IV, CCA sequence, amino acid attaches here. 145. Secondary structure of terna looks like a dash. Clover leaf, clover leaf, clover leaf. 146. In actual structure, terna looks like dash. Inverted L, inverted L, inverted L. 147. There are special terna for stop codons. False, no terna for stop codons. False, no terna for stop codons. False, no terna for stop codons. 148. Amino acids are activated in the presence of dash and linked to their dash. ATP, cognate terna. ATP, cognate terna. ATP, cognate terna. 149. Charging of terna is also called aminoacylation of terna. Aminoacylation of terna. Aminoacylation of terna. 150. The cellular factory responsible for synthesizing proteins is ribosomes. 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 151. Ribosomes consist of dash and about dash no of different proteins. rRNAs and ATRRNAs and ATRRNAs and AT. 152. When large, small subunit encounters mRNA, the process of translation of mRNA to protein begins. Small, small, small. 153. No of sites in large subunit for amino acids to bind is 2, 2, 2. 154. Ribosome also acts as a catalyst for the formation of peptide bond. True, true, true. 155. rRNA in bacteria is the ribozyme. 23 sRNA. 23 sRNA. 23 sRNA. 156. UTR are present only at 5' end before start codon, and not 3' end after stop codon. False. Present both at 5' and 3' end. False. Present both at 5' and 3' end. False. Present both at 5' and 3' end. 157. Dash are required for an efficient translation process. UTR, untranslated regions. UTR, untranslated regions. UTR, untranslated regions. 158. For initiation dash binds to the mRNA at the start codon. Ribosome. 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 159. The mRNA moves from codon to codon along the ribosome. False. Ribosome moves from codon to codon along the mRNA. False. Ribosome moves from codon to codon along the mRNA. False. Ribosome moves from codon to codon along the mRNA. 160. Amino acids are added one by one into polypeptide sequences dictated by mRNA and represented by iRNA. False. Dictated by DNA and represented by mRNA. False. Dictated by DNA and represented by mRNA. False. Dictated by DNA and represented by mRNA. 161. At the end, binds to the stop codon. Release factor. Release factor. Release factor. 162. Tell the 4 levels at which gene expression is regulated in eukaryotes. Gene expression is regulated at I. Transcriptional level, E. Processing level, regulation of splicing, E. Transport of mRNA from nucleus to cytoplasm, IV. Translational level. Gene expression is regulated at I. Transcriptional level, E. Processing level, regulation of splicing, E. Transport of mRNA from nucleus to cytoplasm, IV. Translational level. Gene expression is regulated at I. Transcriptional level, E. Processing level, regulation of splicing, E. Transport of mRNA from nucleus to cytoplasm, IV. Translational level. 163. Beta-galactosidase catalyze hydrolysis of dash into dash and dash. Lactose glucose plus galactose. Lactose glucose plus galactose. Lactose glucose plus galactose. 164. In prokaryotes, dash is the predominant site for control of gene expression. Transcription level. Transcription level. Transcription level. 165. Regulatory proteins can act both activators and repressors. True. 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 166. In a transcription unit, the activity of RNA polymerase at a given promoter is regulated by interaction with accessory proteins, which affect its ability to recognize start sites. Recognize start sites. Recognize start sites. 167. Accessibility of promoter region of dash DNA is regulated by interaction of proteins with sequences termed dash. Prokaryotes, operators. Prokaryotes, operators. Prokaryotes, operators. 168. Operator region is adjacent to the promoter elements in most operons. True, 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 true. 169. In most cases, the sequence of operator binds an activator, repressor. Repressor, repressor, repressor. 170. What is negative regulation? Something is increasing, implies we have to decrease it. Something is increasing, implies we have to decrease it. Something is increasing, implies we have to decrease it. 171. What is positive regulation? Something is decreasing, implies we have to increase it. Something is decreasing, implies we have to increase it. Something is decreasing, implies we have to increase it. 172. The lac operon is shown in diagram. 4. X, in absence of inducer, and Y, in presence of inducer. Identify the genes IV. Label L, M, N and A, B, C, D. Diagram, 4 to 1P, 2I, 3P, 4O, 5Z, 6Y, 7A, L repressor mRNA, MK repressor, N inducer, A, beta-galactosidase, B, permes, C transacetylase, D lac mRNA. Diagram, 4 to 1P, 2I, 3P, 4O, 5Z, 6Y, 7A, L repressor mRNA, MK repressor, N inducer, A, beta galactosidase, B, permes, C transacetylase, D lac mRNA. Diagram, 4 to 1P, 2I, 3P, 4O, 5Z, 6Y, 7A, L repressor mRNA, MK repressor, N inducer, A, beta galactosidase, B, permes, C transacetylase, D lac mRNA. 173. Dash and dash discovered lac operon. François Jacob and Jacques Monod. François Jacob and Jacques Monod. François Jacob and Jacques Monod. 174. Jacob was a biochemist, geneticist and Monod was biochemist, geneticist. Biochemist, geneticist. Biochemist, geneticist. Biochemist, geneticist. 175. Ara operon is for arabinos. 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 176. Regulatory gene is called I gene where I means inducer. False. I means inhibitor. False. I means inhibitor. False. I means inhibitor. 177. I gene codes for dash. Repressor. 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 178. Z, Y, A genes codes for Z, gene, beta galactosidase, Y, gene, permes, A, gene, transacetylase. Z gene, beta galactosidase, Y gene, permes, A gene, transacetylase. Z gene, beta galactosidase, Y gene, permes, A gene, transacetylase. 179. Function of permes and transacetylase are permes, increase permeability of the cell to beta galactosidase, transacetylase, transfer acetyl group to beta galactoside. Permes, increase permeability of the cell to beta galactosidase, transacetylase, transfer acetyl group to beta galactoside. Permes, increase permeability of the cell to beta galactosidase, transacetylase, transfer acetyl group to beta galactoside. 180. Inducer for lac operon are lactose and allolactose. Lactose and allolactose. Lactose and allolactose. 181. A very low level of expression of lac operon is present in the cell always. True. 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 182. The repressor of lac operon is synthesized all the time. True. 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 183. The repressor is inactivated by interaction with inducer, like lactose or allolactose. Inducer, like lactose or allolactose. Inducer, like lactose or allolactose. 184. What are constitutive genes? Those genes which constantly express themselves in a cell. Those genes which constantly express themselves in a cell. Those genes which constantly express themselves in a cell. 185. Regulation of lac operon is positive, negative. 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 186. HGP was launched in year 1990. 1990. 1990. 187. HGP was closely associated with dash. Bioinformatics, 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 bioinformatics. 188. Approx. How many genes are in human DNA? 20,000, 25,000. 20,000, 25,000. 20,000, 25,000. 189. ELSI full form. Ethical, legal and social issues. Ethical, legal and social issues. Ethical, legal and social issues. 190. HGP was a year-long project coordinated by Dash and Dash. 13. U.S. Department of Energy and National Institute of Health. 13. U.S. Department of Energy and National Institute of Health. 13. U.S. Department of Energy and National Institute of Health. 191. Dash became a major partner in the early years of HGP. Dash. Wellcome Trust, UK. Wellcome Trust, UK. Wellcome Trust, UK. 192. HGP was completed in year 2003. 2003. 2003. 193. Nematode whose genes are sequenced is Cynohabditis elegans. Cynohabditis elegans. Cynohabditis elegans. 194. It is pathogenic, non-pathogenic. 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 195. Plants whose genes are sequenced. Rice and Arabidopsis. Rice and Arabidopsis. Rice and Arabidopsis. 196. Name the non-human models whose genes are sequenced. Bacteria, yeast, Cynohabditis elegans. Bacteria, yeast, Cynohabditis elegans. Bacteria, yeast, Cynohabditis elegans. 197. Est full form. Expressed sequence tags. Expressed sequence tags. Expressed sequence tags. 198. The two methodologies for HGP were ESTN sequence annotation. ESTN sequence annotation. ESTN sequence annotation. 199. For sequencing the genome of an organism, when a geneticist focuses on identifying all genes that are expressed as RNA, this methodology is called EST. EST. 200. For gene sequencing, when a geneticist uses a blind approach by simply sequencing the whole set of genome that contains all the coding and non-coding sequence and later assigning different regions its function, this methodology is called sequence annotation. Sequence annotation. Sequence annotation. 201. For sequencing, DNA is converted into fragments and cloned because result into amplification of each piece of DNA so that it can see sequenced with these. Result into amplification of each piece of DNA so that it can see sequenced with these. Result into amplification of each piece of DNA so that it can see sequenced with these. 202. Commonly used hosts for cloning are bacteria and yeast. Bacteria and yeast. Bacteria and yeast. 203. BEC and YAC are dash back and YAC full form are vectors, bacterial artificial chromosome and yeast artificial chromosome. Vectors, bacterial artificial chromosome and yeast artificial chromosome. Vectors, bacterial artificial chromosome and yeast artificial chromosome. 204. The DNA fragments were sequenced by the method developed by dash Frederick Sanger, Frederick Sanger, Frederick Sanger, Frederick Sanger. 205. Dash is credited for developing method for determination of amino acid sequences in proteins. Sanger, Sanger, Sanger. 206. Last chromosome to be sequenced was dash which was sequenced in dash chromosome I, May 2006. Chromosome I, May 2006. Chromosome I, May 2006. 207. Human genome contains dash million BP. 3164.7. 3164.7. 3164.7. 208. Average gene consists of dash no of bases. 3000. 3000. 300. 209. Largest known human gene is dash at dash no bases. Dystrophin, 2.4 million. Dystrophin, 2.4 million. Dystrophin, 2.4 million. 210. Dash percent of nucleotide bases are exactly the same in all people. 99.9. 99.9. 99.9. 211. Less than dash percent of the genome codes for proteins. 2%. 2%. 2%. 212. The functions are unknown for over dash percent of the discovered genes. 50. 50. 50. 213. Repeated sequences make up very large portion of the human genome. T. 214. Dash chromosome has most genes with dash no of genes. Chromosome 1, 2968. Chromosome 1, 2968. Chromosome 1, 2968. 215. Dash chromosome have least genes with no of genes. Y chromosome, 231. Y chromosome, 231. Y chromosome, 231. 216. In about dash million locations SNPs occur in humans. 1.4. 1.4. 1.4. 217. DNA fingerprinting involves identifying differences in some specific regions in DNA sequence called as repetitive DNA. Repetitive DNA. Repetitive DNA. 218. In repetitive DNA, small stretch of DNA is repeated many times. True. 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 219. Small peaks during density gradient centrifugation are referred to as dash DNA. Satellite part of repetitive DNA. Satellite part of repetitive DNA. Satellite part of repetitive DNA. 220. Satellite DNA is classified as micro and mini satellites on the basis of base composition, at rich or GC rich, length of segment, and number of repetitive units. Base composition, at rich or GC rich, length of segment, and number of repetitive units. Base composition, at rich or GC rich, length of segment, and number of repetitive units. 221. DNA from every tissue from an individual show different degree of polymorphism, F. Same degree of polymorphism, F. Same degree of polymorphism, F. Same degree of polymorphism. 222. Polymorphisms are inheritable from parents to children. True. 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 223. DNA polymorphism forms the basis of dash and dash. Genetic mapping and DNA fingerprinting. Genetic mapping and DNA fingerprinting. Genetic mapping and DNA fingerprinting. 224. Polymorphism arise due to mutation. 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 225. If more than one allele occurs with a frequency greater than dash, it is called DNA polymorphism. 0.01. 0.01. 0.01. 226. Probability of such variation in non-coding DNA sequence is more. True. 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 227. There is a variety of different types of polymorphisms ranging from single nucleotide change to very large-scale changes. True. 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 228. For evolution and speciation, such polymorphisms do not play any role. False. 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 229. Technique of DNA fingerprinting was initially developed by Alec Jeffreys. Alec Jeffreys. Alec Jeffreys. 230. He used bulk DNA, satellite DNA, satellite DNA. Satellite DNA. Satellite DNA. 231. VNTR full form. Variable number of tandem repeats. Variable number of tandem repeats. Variable number of tandem repeats. 232. VNTR involved northern blot hybridization, F. Southern blot is involved, F. Southern blot is involved, F. Southern blot is involved. 233. In blotting, DNA fragments are transferred to membranes made of dash or dash. Nitrocellulose or nylon. Nitrocellulose or nylon. Nitrocellulose or nylon. 234. Southern, northern and western blot are respectively used for DNA, RNA, protein. DNA, RNA, protein. DNA, RNA, protein. 235. Detection of hybridized DNA fragments is by dash. Autoradiography. 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 236. Describe the 6 steps of DNA fingerprinting. Steps of DNA fingerprinting: I. Isolation of DNA. E. Digestion of DNA by restriction endonucleases. E. Separation of DNA fragments by electrophoresis. IV. Blotting of separated DNA fragments. V. Hybridization using labeled VNTR probe. V. Detection of hybridized DNA fragments by autoradiography. Steps of DNA fingerprinting: I. Isolation of DNA. E. Digestion of DNA by restriction endonucleases. E. Separation of DNA fragments by electrophoresis. IV. Blotting of separated DNA fragments. V. Hybridization using labeled VNTR probe. V. Detection of hybridized DNA fragments by autoradiography. Steps of DNA fingerprinting: I. Isolation of DNA. E. Digestion of DNA by restriction endonucleases. E. Separation of DNA fragments by electrophoresis. IV. Blotting of separated DNA fragments. V. Hybridization using labeled VNTR probe. V. Detection of hybridized DNA fragments by autoradiography. 237. VNTR as a microsatellite, minisatellite, mini-satellite. 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 238. The size of VNTR varies from dash to dash basis. 120,000. 120,000. 120,000. 239. The autoradiogram will give bands of same sizes in monozygotic twins. Monozygotic twins. Monozygotic twins. 240. The sensitivity. This technique has been increased by use of PCR. PCR. PCR.